Okay, now we're going to look at the principles of infectious disease and epidemiology. This is found in your text in Chapter 9. It is Lecture 8, and so we'll take off with it. Now, there are many different causes of infectious disease, but we have to get down the terminology, as that's the foundation of understanding modern healthcare and epidemiology. So when you talk about terms, let's go through this. Infectious disease, this is an illness caused by a pathogen. Opportunistic pathogens are only cause disease when the host, the targeted organism, is weakened in some way. For example, it could have shifts in normal microbiota or a weakened immune system. Now, many people, when they hear opportunistic infections or opportunistic pathogens, think of HIV patients, but you have to consider beyond that. You have many individuals that are on certain types of cancer therapy, that their immune system will be also blunted. Individuals that have received tissue or actual organ transplants and may be on anti-rejection drugs, which act to blunt the immunity as well. Now, a true pathogen doesn't require a weakened immune system of its host to cause disease. When we talk about sporadic cases, this is disease of isolated infections in a particular population. When we talk about endemic infections, these are routinely detected in a population or region. For example, if you went to, let's say, some southern area of Africa, an endemic infection might be malaria. You would not find it in Alaska. If you started seeing this in Alaska, that would be room for concern. Now, an epidemic is a widespread disease outbreak of a particular region during a specific time frame. And in a case of an epidemic might be, for example, there was an outbreak of Nipah virus, which has a 40% mortality rate in Malaysia, all right? And that was quite significant. So you would have it spread throughout the population. Once individuals started getting infected, they would basically spread it to others. And then you would have individuals that are sick that would be in that particular limited area of, we're talking country, province, or if you really want to go further, one of the continents. Now, emerging pathogens are a newly identified agent or pathogens that previously caused sporadic cases, but are now increasingly common and or exhibit an expanded geographic distribution. Case in point, Ebola virus. The Ebola virus was first isolated in Ebola, which was the Ebola community, which I believe was in Uganda, in Africa. 1976. Nobody had really seen that, and then it died down. And for about several decades, they really hadn't had any more incidents. A little bit here, a little bit there. But then what really happened is once we got into the 21st century, we had a widespread outbreak that occurred on West Africa, which spread beyond the borders of one or two countries. And so it was becoming increasingly common, and it had expanded its geographic distribution. And because of the global, basically the global capability to go from one continent to another, or from one side of the planet to the other in less than a 24-hour period, that's where it got very scary for the possibility that it could expand beyond the original geographic distribution. Now, a re-emerging pathogen is an infectious agent that was under control due to prevention or treatment strategies, but now is resurfacing. A case in point, situations where a re-emerging pathogen might occur might be malaria. And malaria in certain parts of the world was basically kept under control due to prevention or treatment strategies. For example, spraying areas where mosquitoes were, et cetera, or the use of certain drugs that basically prevent the life cycle of malaria in humans, such as chloroquine, primaquine, hydroxychloroquine, etc., as well as artemisinin. Now, somehow that control broke down. In certain areas of the world, malaria has resurged, and this is in part, its resurfacing was caused because the malaria parasites are now resistant to chloroquine, primaquine, artemisinin, etc., these drugs. Now, a zoonotic disease is an emerging infection spread from animals to humans. Case in some people's thought is either bird flu or swine flu, or even COVID-19, where it was believed that COVID-19 originated from either bats, may have gone through an intermediate stage with pangolins, and then eventually jumped over into humans and became communicable by humans. With bird flu, it was an influenza strain that jumped from the birds over to humans. Same thing with swine flu, that the influenza was prevalent in swine and then was able to jump over, usually through antigen shifts, and it became infectious for humans. Now, when we say something's non-communicable, it's a disease that doesn't spread from person to person. Okay, what's an example of that? Anthrax. Anthrax, yes, because when someone gets anthrax, they get the anthrax bacteria into them. It's not as if you have a patient who has anthrax could spread it to other patients in the ward or something else like that. This is a disease that has to spread by the bacteria. They either have to be inhaled, consumed from contaminated meat, or basically picked up by open cuts, etc., where the bacteria get into the wound. But it's not something where basically you could sneeze and give your neighbor anthrax. Communicable, though, these are diseases that transmit from person to person. Now, a lot of times what you have to understand is that mutations in the pathogen can induce a non-communicable disease to become communicable. That's one of the problems we have. We've talked about before antigen drift and antigen shift. It's going to be much more basically on the table as we go through this discussion, because this is a very serious situation that can make a disease pop up and suddenly become a worldwide threat. Now, descriptors of an infection as experienced by the host. Well, how do you basically say, Doc, I don't feel well? Well, what are your signs? What are your symptoms? You see, when we have an active infection, the patient is symptomatic of the disease, meaning they have signs and symptoms. Signs are objective indicators of the disease that can be measured or verified. Example, fever, a rash, blood in the stools. Symptoms, though, are factors experienced by the patient and are subjective rather than precisely measurable. Now, how many of you have probably seen the situation where you hear someone say, okay, are you in pain? Yes. On a scale of 1 to 10, 10 being the highest, what is your pain? Some people will say 2, some say 3. Some will basically have a certain level of pain and it's become unbearable. So they might say 7, 8, 9. So there's no real, how I can say, pain meter that they can attach to somebody and say, well, you're in this much pain, okay? It's all subjective to the patient. Same thing with fatigue, and the same thing with nausea. Now, latent infections, the host does not have the signs or symptoms, hence they're asymptomatic. With COVID-19, let me go back to that virus for a second. We've had cases where patients, before they actually showed the symptoms, were themselves infected and were shedding the virus. This is something that is very real in many other diseases, not just the COVID virus. Now, infections can be described by time, either the onset of the actual illness or the duration in which a person is ill. If we talk about acute disease, that's a rapid onset and the progression of disease shows up. So you're exposed within 1-2 days, boom, you've got it. Give you a quick example. Love my daughter. But when my daughter was 4 or 5, she had what a lot of kids have, and they ended up with was like an upper respiratory infection. But like all kids, sometimes they don't remember immediately to put their hand over their mouth when they're coughing. So I held my daughter up close, and she went, and of course, it was right into my open mouth almost. And yes, in 36 hours, I was sick. A chronic disease, though, differs. It's got a slower onset and progression. Some of this is based on the way the organism fights against the host immune system, as well as how the organism may incubate within the body. Okay? So there are a variety of factors there that differentiate chronic disease from acute disease. Now, there's 6 groups of pathogens. Some of you have been reviewing some of these as we go through the labs. Helminths, protozoans, fungi, bacteria, viruses, and prions. Now, some of them have differing levels of severity, onset, etc. And we'll try to go through some of these as I go through the entire concept of infectious disease and epidemiology. Now, we have to go back to this concept that we dealt with at the early part of the course, Koch's postulates. Robert Koch came up with this concept that basically reveals sort of a cause and effect, the cause of some infectious diseases. If you remember, basically it's the same organism must be present in every case of the disease. Organisms must be isolated from the diseased or host and grown as a pure culture. Isolated organisms should be the cause of the disease in question when it's introduced, that is inoculated, into a susceptible host. You got to keep in mind that this is an important point, a host that can develop the disease. The organism must be then re-isolated from the inoculated diseased animal. Now, that sounds pretty straightforward, but there are some caveats, warnings. For example, the postulates don't always apply to non-infectious diseases, illnesses not directly caused by pathogens. Next, you have certain organisms that are very tough to culture, okay? For example, obligate intracellular pathogens, Rickettsia, viruses, certain protozoa. They will only replicate inside of the host cell, and hence they are difficult, if not impossible, to grow in independent pure cultures. You will find that there are some that are very difficult to culture. And what happens is you have to use another related pathogen to get an idea of the history and how this pathogen develops and causes disease. An example of this, of course, is Treponema-pallidum, which is the pathogen that causes syphilis. They don't have a really good syphilis culture plate or anything like that. So they have to culture the organism in rabbits. Rabbits actually have a related strain of syphilis that's passed from bunny to bunny to bunny. Yes. Now, the point being, you can use similar drugs to try to eradicate the syphilis of the bunnies with reasonable assurance that you can do the same thing with humans. Also, you have some organisms that its growth in a pure culture can result in attenuated pathogen. When I say a pure culture, by the way, this is when we get into in vitro versus in vivo. In vivo means in a living organism. In vitro, such as growing it in tissue culture, growing it on a petri dish, this is called in vitro, as if in glass. Yeah, they used to have petri dishes that were made of glass. Now they're disposable, they're made of plastic. But the problem that you have when you use some culturing techniques is that after a couple of generations of the organism, you end up with a non-disease causing or at least a weakened one. Oh, by the way, one other example of what I was just mentioning is HIV. They could not grow in the beginning HIV easily. And so they had to look at and use studies of a related form of HIV that was unique for primates. This was simian immunodeficiency virus. Okay. And they got a lot of research out of that because if you infect, interestingly enough, HIV, human immunodeficiency virus, into the primates, the virus will culture, but it won't cause them to have sickness or immunodeficiency, immunosuppression. Okay, let's keep moving. Now, if you look at infectious disease, here is one of our nasty little bacterias, Bacillus anthracis. This is the pathogen for anthrax. Notice that you see the endospores forming in here in the bacteria. Now, we're going to start dealing with infectious disease transmission and stages. Pathogens can come from different sources, some of which are reservoirs. A reservoir, animate or inanimate habitat where the pathogen is naturally found. Let's go over a few examples. Leprosy, Mycobacterium leprae. This is actually found in 9 banded, it's called band armadillos. Now, these are out in the southwest. They are armadillos. They have 9 particular stripes or parts of their plating on the exterior of their body, but they very comfortably have leprosy in their body, and it doesn't cause any effects to the armadillo. If humans come in contact, it would cause serious problems. Lyme disease, Borrelia burgdorferi. Its reservoir is the white-footed mouse. Chagas disease. This is a protozoal parasite called Trypanosoma cruzi. And it's interesting in that it can be found in domestic dogs. I could even take it further. You can find COVID and COVID-relevant viruses in bats. The source, a lot of times, will disseminate the pathogenic agent from the reservoir to new hosts. This is where we also get into an interesting little concept called vectors. This is an organism, for example, a tick, fly, a mosquito, that transmits a disease or a parasite from one animal to another. Very commonly, if you go back to Lyme disease, well, we know that that's caused by a tick bite. Malaria, well, that's caused by the bite of an infected mosquito. And flies, well, flies can carry all sorts of bacteria on their feet, etc. I've included that in a subsequent lab that you're going to deal with later on. So it's important to know the source of the infection as well as the reservoir. Endogenous sources, these are where the pathogen came from the host's own body. An exogenous source is a source that is external to the host. So let me go over a couple of better examples to help you with this. Exogenous sources, for example, the environmental, could be contaminated food, water, contaminated soil, medical equipment. When we talk about animals, it could be animals that transmit zoonotic diseases to humans. If it's humans, they could transmit communicable infections from one person to the other. When we talk about endogenous sources, this is a situation where you have misplaced normal microbiota. Now think about it this way. We have bacteria living on our skin, doesn't cause any illness, but you have an opening of the skin, let's say due to a surgical incision, and it leads to eventually a post-operative infection. That's the same bacteria. But now they're not on the skin. They're in that wound, and they will proliferate and cause disease. Also, we have disrupted microbiota and opportunistic infections. For example, yeast in the vagina may proliferate and cause infection after antibiotics kill off the bacterial neighbors. Many women suffer yeast infections after they've been given an antibiotic treatment. How does this occur? In the vagina are the natural microbiota, and these include certain lactobacilli bacteria that make lactic acid. As long as they are there, they create a lower pH, which makes the environment hostile to the proliferation of yeast. Once they're eliminated by the use of antibiotics, the yeast may then rebound and proliferate in the vagina. Okay? Now, transmission is really the spread of a pathogen from a source to a new host. We have direct contact transmission. The host comes into physical contact with the source of the pathogen. Well, what's the contact? Well, it could be swimming, soil, bite of an animal, bite of a mosquito, bite of a greenbottle fly. It could be saliva. It could be sexual intercourse. For example, why is it risky to swim in a warm pond or lake water? Now, this is warm, unagitated water. Let's say it's in the summer. This is very common where you will have Naegleria fowleri. This is a amoeba that proliferates in some of these warm waters. But when one swims in, particularly dives nose or head first, they force the water up into the nasal cavity, where some of these amoeba will follow up the olfactory epithelial cells and follow up through those olfactory axons up through the cribriform plate and then proliferate into the brain. Once this infection has occurred, death usually occurs in about 10 days. There's been very limited success in attempting to treat someone successfully with any type of antiprotozoal drugs, and it's pretty sad. It's very common in the southern states to have this occur, much less common in northern states where the waters are usually cooler and you don't just have this happening. Now, there's another concept I want to get you to understand. It's called vertical transmission. This is specialized direct contact where the pathogen passes from mother, there was a typo there, sorry about that, mother to the offspring during pregnancy or during delivery or during breastfeeding, okay? Indirect contact transmission, this is where the pathogen spreads without direct physical contact with the source. So when you have indirect contact, this is where you're going to have all these situations like airborne transmission. The pathogen enters through the respiratory route, usually inhaling pathogen-laden respiratory droplets expelled by coughs or sneezes. Now, be aware of something here. There's a term called the route, excuse me, portal of entry. The portal of entry of many pathogens can be the nasal or the oral area. But as we're seeing data coming out, COVID-19, another portal of entry is the eyes. Normally, a lot of pathogens are neutralized by the lysozyme in the tears, but it appears that in some documented cases, the portal of entry was the eyes, and this led to some very severe illnesses because of the COVID-19 virus. Now, vehicle transmission. This is also known as formite transmission. This is the pathogen is introduced via contaminated inanimate objects. Okay, this could be doorknobs, door panels, blankets, needles, food, water. Wherever the contamination went to these particular items, the person then comes in contact with the items, and that's how they pick up the pathogen. Now, when I talk about vector transmission, I've mentioned this before. These are organisms that spread infectious agents to other susceptible hosts. There are two types of vectors. A biological vector. This is where the vector organism has a role in the pathogen's life cycle. So to help you understand this, think of the mosquito, think of malaria. The mosquito, and it's, by the way, only the females that are doing this, because the females have to have a high protein meal for their eggs. So the mosquito comes in, sucks up some blood from a human being. As it sucks up the blood, it's taking in some malaria parasites from the human being's blood as well. The blood, of course, is broken down. The malaria parasites will basically gestate inside of the midgut of the mosquito. As a result, they will go through a round of the life cycle, and then the new offspring will migrate up to the salivary glands of the mosquito. The next time the mosquito bites someone else, what the mosquito will do is it will inject some of its saliva. That's what accounts for somewhat the itchiness afterwards with a mosquito bite. But the saliva contains anticoagulants that prevent the bite site from basically clotting a little bit. But also what will be in that saliva are the malaria parasites that start the cycle again within the human being. I hope that's made clear. Now, mechanical vector, these are a little bit different. They spread disease pathogens without being integral to the pathogen's life cycle. So it's either on the external part of the body, it's on the feet. For example, flies. Flies will very commonly have on their feet a whole host of different types of pathogenic bacteria. They'll go from a pile of dog feces to your dinner table. They will go from the sandbox where animals have done their business in it, and then they will move over to the picnic table and onto the sandwich. I know that's not appealing, and I know it may be somewhat grotesque for some people to think about, but this is how we get contamination, and this is how the fly acts as a mechanical vector. This chart here gives you some ideas about the various types of direct contact transmission and the various types of indirect transmission. And you can see that person to person, animal, environment, vector. With indirect, of course, we've got airborne, we've got vehicles. One of the vehicles is contaminated needles, but it could be also contaminated water. Cholera has been known to be transmitted by either contaminated water or contaminated food in some studies. And then, of course, we have the vectors, mosquito bite, tick bite, flea bite. And then we have the mechanical vectors, which are basically hitching a ride. As you can see there, one other example, cockroaches. They scurry through garbage and then through food. They are very common to have salmonella bacteria on their bodies. And then, of course, that's how they spread it to the human population. Now, there are 5 general stages of disease which occur during infections. I want you to get used to some of the terms, and I want you to be ready to handle all the 5 general stages. For example, infectivity. This describes how effective a pathogen is at establishing an infection. Virulence describes the severity of the disease following an infection. Pathogenicity, this is the general ability of an infectious agent to cause disease. Now, there are generalizations and variations exist between patients as well as between pathogens. This is not to say it's all loosey-goosey, but you have to understand that there is a little bit of variability between how pathogens interact with individual cases. Also, it's possible for diseases to be transmitted to other susceptible hosts at any point in these 5 stages. This is extremely important to note, since when we started dealing with, let's take COVID-19, we a lot of times saw individuals shedding the virus, not feeling bad, but later on subsequently feeling ill. So in other words, there was a point in which the person was having the virus build up inside of the individual, but they weren't feeling sick yet. But at the same time, they were basically a carrier to spread the disease to other individuals. So let's go through the stages. First off, we have the incubation period. This is the time between infection and development of the disease symptoms. Now, the next is the prodromal phase. This is where the early symptoms develop. You want to keep in mind that after prodromal, the patient may develop an acute phase, experience the full-blown classic symptoms of the disease. You will also have to be aware of asymptomatic cases. These are the subclinical cases. The symptoms are mild to non-existent. The pathogen may exist for extended periods without experiencing any symptoms. Does this happen a lot with COVID? There are cases that are reporting it, although there is some dispute on this. But there have been individuals with other diseases that were able to pass on certain viral diseases or bacterial diseases before they really got sick themselves. And unfortunately, by that time, when they finally became noticed, you had to literally track down all of the persons that were exposed. This is known as contact tracing. Now, we get into the acute phase, that's the peak of the disease. Then we have a period of decline. This is the replication of the pathogen is brought under control. Usually you have finally the rallying of the immune system that plays a role in this, and the symptoms begin to subside. Now, if on antibiotics, the patient may not fulfill treatment, and hence the pathogen may develop antibiotic resistance. That's why your doctors are always telling you, make sure you complete all of your antibiotics, even after you start feeling better. Because what happens is that with the dosage that may exist, if certain pathogens are able to survive a smaller amount, they may develop the resistance and come back to make the patient ill again. Finally, we have a convalescent period. This is a period for patient recovery. The pathogen is basically eliminated, but the host may harbor the virus indefinitely. The pathogen may be kept in a latent state in the patient. Case in point, many of us may have gotten chickenpox. But if you did get chickenpox, it's entirely possible that the unique herpes zoster virus, which causes the chickenpox, may be lying dormant in the trigeminal ganglia and in other neural ganglia in the body. When the immune system basically gets suppressed, or when the individual's immune system is reduced, such as under stress, then you may have the resurgence of this virus now coming back. That's what we call shingles. So, incubation period, you can see this is the time between infection and development of early symptoms. Prodromal, early symptoms develop. Acute phase, the peak of the disease. Period of decline, the replication is brought under control. Symptoms start to resolve. Then the convalescent phase, this is where the patient recovers. In some cases, the pathogen may be kept latent in the patient, so that's what you have to be mindful of. Now, we have these situations where we talk about subclinical cases of the disease. And by age, basically, the different types of organisms may exist, but it really depends on several of the factors. There are no real, if you notice at the bottom, subclinical cases for rabies. If you get it, it's fatal. There are some strategies that are being developed now and have started to build up to treat someone with active rabies. But the idea is that you want to treat the person regardless of whether or not they had it. If they were possibly exposed, then you immediately deal with them with a rabies vaccine. Because once they have the rabies, it's a very quick downhill course for most patients. But what am I trying to say here? If you notice the numbers and notice the different diseases, what you have are situations where people might feel, oh, I didn't feel well, I was tired. Oh, I didn't feel this, I didn't feel that. But as time goes on, they won't get any worse. But as a subclinical case, they may be a spreader of that particular pathogen. And that's why you also want to be mindful of this. Now, here's a classic case of someone who was, and in the modern parlance, we would call it a super spreader. What this person, and who this person was, she was referred to as Typhoid Mary. Her name was Mary Mallon. She lived between 1869 and 1938. I'll give you the story about Mary. Mary was a illiterate. She was an Irish immigrant, but she was a very, very good cook. And all she really knew was to cook. She was what is referred to as an asymptomatic carrier of Salmonella enterica typhi. Usually this is a type of salmonella bacteria that will colonize the gallbladder, and it will be excreted. And usually at that time, people didn't wash all their hands absolutely and everything else like that. But Mary would shed salmonella bacteria. And what happened was it was the first case found by some public health doctors in the, I believe it was on the Manhattan Island, or it could be on the Long Island. The big problem they had was, where's this coming from? Because they didn't have any other prior outbreaks, but it was a certain grouping of people that started all having reports of typhoid. Eventually, they boiled it down to Mary. So the doctors went in and said to Mary, look, you got to do something else. You cannot be a cook. And she unfortunately would say, look, this is all I know how to do. So they actually had someone teach her sewing so she could have basically a means to support herself. So a couple of years went by, and then in another unusual sort of like country estate, almost like a country club, they started having another outbreak of typhoid. So what happened was the doctors walked into the kitchen, and one of the doctors immediately looked, and there was Mary at it again, cooking, and they immediately seized her and arrested her. Now, you have to understand something. She had broken the agreement. She had not done very well with sewing, and so she went back to what she knew best, cooking. As a result, the public health authorities went before the judge and said, we can't have her roaming around, spreading typhoid everywhere. And so by court order, she was quarantined onto an island near Manhattan. And she lived there until she was of a certain age. She passed away basically in 1938. But they couldn't do anything. They had to basically keep her, in essence, in quarantine, in a jail, if you want to say, because she was a public hazard, a public health hazard to anybody that she served food to. Okay. Now let's talk about some epidemiological essentials. Again, you got to know the terms. When we talk about epidemiology, this is the branch of medicine which deals with the incidence, the distribution, and the possible control of disease. Keep all three of these concepts in mind. It's not just tracking down where a particular disease is occurring. What are the general goals of epidemiology? To describe the nature, cause, and extent of new or existing diseases in populations, to intervene to protect and improve health in populations. Now, there is the renowned epidemiological triangle, which I'll show you in a diagram in a second. It's also known as the disease triangle, which links the host, the etiological agent, basically the pathogen, if you want to say, and the environment together. Now, when you have those all together, this gives you a good idea about how and why and when diseases occur. There's further extension of this called the disease pyramid. The disease pyramid was more set up for plant disease specialists, but it applies equally, if not better, to all epidemiology, not just phytoepidemiology. The disease pyramid, think of it first, I had a triangle, so I had 1, 2, 3 sides, but now with the pyramid, I've got basically 4 sides. And it links the host, etiological agent, environment, and the other key point, time. So this is sort of a remnant of this. When I talk about environmental factors, what are these? Climate, geographical location, availability of the transmitting vector, water source, food source, etc. Okay? Now, if you think about that, just take malaria. Like I said, it sounds crazy, but if I ever heard someone say, look, you're going to have malaria outbreak in Juneau, Alaska, be like, why? How? There's no mosquitoes. There's tons of mosquitoes, but they're not the mosquitoes that transmit malaria up there. Etiological agent could be a fungi, bacteria, virus, or parasite. The host factors, their general health, the sex, lifestyle, age, ethnicity, occupation, etc. Now that's the disease triangle here. So you have one side being pathogen, one side the host, one side the environment. And you have to consider all of these points. What's the condition of resistance? Is it a predisposed situation, or is it, for example, because the person becomes older? What's the condition of the pathogen, the virulence, the domain, the population? What environmental conditions does it require? Okay. That's the pathogen. And then the environment. This includes, well, for foliar diseases like plants, moisture, temperature, soil diseases, pH, etc., texture. Here's one way to think about it for human beings. Host, pathogen, the environment. Now, let's think about that for 1 second. Take influenza. When do we see it more often? Well, fall going up in through the winter months, dying out by about spring. True. If you had a Shigella outbreak, which is usually one of these fecal-oral transmission routes, when would you see it more often happen? Well, during the summer months. You know, when people are outside, if maybe they don't wash their hands as much, etc., in the warmth and so on and so forth of the summer months, that's when you're going to have this. You could include it for environment if you were going to say in a high population density, such as a daycare center. And that's another example of environment. Okay? Now, here is the disease pyramid. Environment, plant, pathogen, time. Keep in mind, time happens to play an important role. Well, how? Because sometimes, with some diseases, it's not you're exposed and the next day you're sick. Smallpox, you could be exposed, and it may take on average up to about 14 days before you start showing the signs of the illness. With situations like chikungunya, it's a 7-day situation. With COVID-19, it may be 7 days or more. On average, in the beginning, it was said to be about 21 days. Now it's believed to be about 2 weeks. So that's why we have people go into quarantine, they wait 2 weeks, and if they're not still feeling okay, then they come out from quarantine. Now, there are strategies to break the epidemiological triangle, and we're going to focus on the epidemiological triangle only. First, public education. Sometimes you have to inform and teach the population of situations where diseases can be spread. Here's a couple of examples. In the Maasai tribes, what they would do, because cow manure was so important and related to their agricultural culture, etc., individuals, when they were born, were given a little bit of cow manure on the stumps of their umbilicus. The problem is that same stuff would transmit tetanus. And so you had to finally break that cultural connection. The Foray tribe of Papua New Guinea, they had a prion-based disease called Kuru. Handling and consumption of dead relatives and Kuru infections was connected. Basically what happened is when someone dies, in the Foro tribe, as part of the, if you want to say, funeral arrangements, the individuals would handle the deceased's brain, and in some cases, take small pieces and consume them. And they basically passed this around to all of the people that visited. The problem was that you were passing around the brain that was infected with Kuru, and basically had the prions in there, and they would spread it around to pass on as the generation for an infection for the next generation. When the Foray tribe stopped doing that particular custom, the incidence of Kuru virtually disappeared. Now, with India and the United States, we have to teach individuals about the proper use of antibiotics and the spread of antibiotic resistance. And many of you are probably familiar with how hard it is in the United States. Even some people who have clear signs of a viral infection. No, doc, cough, cough, hack, hack, hack. I still want antibiotics. And the problem is, decade after decade after decade, we've been talking about this to patients. They either ignore it or insist on antibiotics. And then what happens? We now have strains of bacteria infections that are resistant to various types of antibiotics. Next issue, quarantine. This is a period of confinement away from the general population for animal or human that may have contracted the disease. Quarantine actually comes from a Venetian variant of the word for 40 days. What would happen is in the 17th century, when a lot of trade was occurring in Venice with the Venice traders and all of these very rich Italian power brokers, I might call them power brokers today, but basically they were tradesmen, etc. What happened if a ship came into the harbor and there was a disease on board, let's say smallpox or plague? The ship would not be allowed to disembark, and the ship would be told you have to stay in the harbor. Nobody leaves the ship for a period of 40 days. That would basically prevent the disease from spreading, although in some cases it led to most of the ship's passengers and crew dying off in the process. But this was to prevent the disease from spreading to the general population that was at the harbor. Now, the other issue that you have to deal with today in quarantine is issues of freedom of movement versus civil rights versus public health. We've seen this with COVID-19. We've seen this when we had the situation with SARS, which was around 03, 05, 06, where the precursor, which was also a coronavirus, the severe viral disease of SARS, what happened was a couple of individuals came to Toronto, Canada. When these individuals exposed others, there was one particular individual that was in a hospital at Toronto. Now, the Minister of Health came into the hospital and said, everybody's got to stay here. You can't leave. And literally, one individual said, no, I'm going to leave. You can't stop me. Bleep, bleepity, bleep, bleep, bleep. And he left. He went back to his home to see his partner, and eventually basically came back to the hospital and eventually was sort of arrested and detained. The problem is that individual went out and spread the potential of SARS to the entire community of Toronto. For one weekend, all of Toronto stopped. No one was allowed to go anywhere. Everyone was told to stay in their homes for fear that they could spread this disease. And when the public health authorities thought it was safe, they basically said fine. But it was a very eerie sight. I remember seeing news stories where you would look at what would have been normally a very busy Saturday in Toronto, and no one was on the streets, on the roads, at the markets, at the restaurants. Everything was like a scene from the Twilight Zone. Now, a third means to help control some of these diseases is vector control. So you would control the biological vectors, which basically helps prevent the spread of disease. When we talk about arboviruses, these are really, it's sort of a shortened name for arthropod-borne viruses. Remember, arthropods include, well, all the insects, okay, as well as all of the arachnids. So that includes, ticks as well as flies, fleas, mosquitoes, cockroaches, and we can go on and on and on. But when we usually talk with arthropod-borne viruses, usually you're looking at either the flea, the mosquito, or the tick that are best known. And from there, we see West Nile virus, yellow fever, dengue fever, plague, malaria. Now, malaria is not a virus, obviously, and plague isn't, it's a bacteria. But a lot of these diseases are transmitted via these different types of vectors. Controlling the vectors helps control the spread of these diseases. Now, this is an image here of the various disease monitoring stations that exist throughout the United States. Notice there's one in Puerto Rico, in Hawaii, in Alaska. The nearest one in the New England area is Boston. And of course, the headquarters is basically in Atlanta at the Centers for Disease Control. But all of these are basically also testing labs that can speed up the processing of information about a communicable disease and relay some of the data to Atlanta also for final judgment. And in the case of some situations, you may have to move very, very quickly to contain the communicable disease outbreak. Now, public health aims to improve overall health in the population. Essential functions of the public health system include the modern day health monitoring and regulation. So there's sort of a hierarchy of where and how you have the monitoring occurring. Let's start at your own local community, your town or city Department of Health. Those are the frontline people at the local level. They then pass information up to the next level, which in this case of Massachusetts would be the Massachusetts Department of Health. They would also have very qualified labs. They're mostly in Boston, and they do a variety of tests, whether it's something communicable like influenza or COVID-19, but they also do some of the more unusual ones that are bioterrorists, for example, agents, anthrax, smallpox, you name it. This information, though, is still shared with the greater Centers for Disease Control at the national level. And the actual full name for CDC is really the Centers for Disease Control and Prevention. These fall under the U.S. Department of Health and Human Services. So CDC serves as a hub for the central information on epidemiological situations, and basically they depend on communication with the state as well as the local authorities. A lot of times your local departments of health don't have all the testing equipment, but what they do have is the knowledge of reporting information as well as gathering samples that may be forwarded to either the state or to CDC. Now, there's another thing that, and yes, you can access it. It is computerized systems. The internet has provided us with a wealth of information. And yes, I'm talking besides the cute kitten videos. No, I'm joking here, but there is a site, and if you put in PROMED, PROMED, in Google, you'll find it. This is the Program for Monitoring Emerging Diseases. It's one of the largest publicly available emerging disease and outbreak reporting systems in the world. Basically, data is being shared with all of the public health, whether it's international, national, regional, all over the world. And this is kind of important when you think about it, because let's face it, if you jump on a plane, you can go almost around the world in less than 24 hours. Now, if you're in a filtered, recycled air system of a plane, you could cough, et cetera, and basically in the period of less than 24 hours, you can go from, let's say, Lagos, Nigeria, to New York, or to go to Lagos, to London, then to New York, and you could start wildfires of emerging disease outbreaks. So it's important to have this central hub where a lot of information is available that can be shared with basically the rest of the world to maintain sort of the pulse of what's going on, and thereby warn officials or bring to their attention the potential that these diseases are occurring in certain parts of the world and could migrate to other parts of the world. Now, when we talk about epidemiological measures and studies, epidemiological measures are often presented as either ratios, proportions, or rates. When I talk about a rate, for example, measure of an occurrence of an event over time. 5 cases an hour. If you notice with COVID, what has happened? You've had governors and even in some cases local leaders providing day-to-day reports of how many cases of COVID have occurred and how many deaths have occurred. You could also look at a ratio, which is the occurrence of an event in one group compared to other groups, males versus females, senior citizens versus young people, etc. And then we look at proportions, which is the percentage of a whole. So how many people out of, let's say, 100 women get this disorder? How many individuals out of the population? For example, there's some people that basically would say, okay, so this has an incidence of 6 cases per 100,000. And so the numbers look a little different until you start thinking, well, let's see. Let's say we have a population of 1.2 million individuals. Per 100,000, that means that if you had 6, you would have basically about 72 individuals sick in a population of, and I hope I did my math right, 1.2 million if you had a 6 out of 100,000 cases. That looks and sounds different and can be overwhelming and can be underwhelming at times. You have to think about this in the perspective of how the cases are being identified, not only in time, but also are there any other factors? In other words, are all these people coming from a school or from a nursing home or from another area that is open, either open spaced or closed space? You see, the measures of frequency have to include disease prevalence and incidence data. Measures of frequency, they give you the information about the occurrence of a disease in the population over a certain period of time. Cases of COVID, how fast did they start ramping up? How fast did they start going back down? Are they ramping back up again? These are the questions that you have to explore. Now, you have to keep in mind also when I talk about the term population, that is any defined group of people. Is it all young people? Is it all older people? Is it all male? Is it all female? Is it all individuals that are Caucasian? All individuals that are African American? All Latinx? And is it all factory workers? All farm workers? All sorts of different variables have to be considered. When we talk about the prevalence rate, otherwise known as prevalence, this is a measure to describe the morbidity in a given population during a specific time. If you're not familiar with morbidity, basically it means how many get sick. Don't get it confused with mortality. Mortality is how many people die. Now, there's two factors that affect prevalence rate. The incidence rate, the IR of a disease, that's the number of new cases in a defined population in a defined time. So you might say, okay, in the past 3 weeks, we've had, on average, our incident rate has been about 300 a day. Also, we have to look at the duration of the disease. How long does the infection last? For some people, the infection lasts 49 days, meaning that they got sick and they were down for 49 days. If you think about the flu, you might say to yourself, oh yeah, last time I had the flu, I was down for a couple of days. I just slept, and after that, I felt better. Now, we start getting into some of these proportions and ratios, etc. I want you to get somewhat familiarized with them, but don't memorize everything. All right? Unless you want to go into epidemiology, which would be fascinating. If I say TC, that's total cases in a defined population during a given time. TP, that's total people in a defined population during the same time. So if you want to look at things like the numbers here, the PR, which we said earlier, prevalence, you're really measuring the number of cases, comparing it in a defined population by the total people in a defined population. When you talk about TN, that's the total new cases in defined population during a specified time. TS is the total susceptible hosts in the population during the same time. Why is this so important? Some diseases you will see dramatically crash in their numbers after something happens, a new treatment or a vaccine. If you see on this chart, you have the number of measles cases per thousands. But look at how the wiggly line hits where you see the dotted line, and that's where the vaccine was licensed. And suddenly, within a matter of a couple of years, boom, you're well below 100,000. And it's just a pickup every once in a while. And by the way, every one of these peaks is usually due to individuals that become susceptible, that herd immunity, the number of individuals that are vaccinated against this, has diminished so that there's much more availability for the disease to spread. Okay? Now, measures of association may reveal risk factors for a disease. When I say measures of association, what are you talking about? That's the various factors that are linked to the cases of the disease, which may in turn determine who is at risk. Are these all medical workers? Are these all individuals that went to the Lunar Festival, for example, in China? Are these all individuals that were part of a fraternity house? Are these all individuals that basically worked at a particular factory? You have to find out what the variabilities or the association is. Data analysis provides information on disease trends and connections to behaviors, occupations, or exposure risks. And when I said mortality rate, this is the number of deaths during a specific time period. Here, we have an unusual test, and this is the percentage of seropositive for a herpes simplex virus number 2. Now, this is important, HSV2. What you see are individuals, and they break them down to white, non-Hispanic, Mexican American, black, non-Hispanic. And notice that the bar is up and down here. They reflect about a 95% confidence interval. But you have a histogram here of basically periods or groupings over a certain period of time. In this case, the groupings are how many sexual partners these individuals had. 1, 2 to 4, 5 to 9, 10 or more. And what you can keep seeing is this what? Increase in individuals. The more partners they have, the greater the risk that they're going to be seropositive, meaning they were exposed to the virus. They now have antibodies against HSV2. You have to be mindful of one other thing. See that first line there? Correlation does not imply causation. Oh, okay, so let's see. I got up this morning, the sun came up, so because the sun came up, I got up. Or I heard a bird tweet. So when the sun comes up, the bird tweets. Really? And just because something occurs at the same time doesn't necessarily connect them to the disease. This is part of the problem. We have to be willing to review if there really is a connection. It may be that they're completely independent from each other. They do occur at roughly the same time, but they're not in any way connected. And that's part of what we have to do at times. Now, there are a variety of mortality rates that you need to be a little bit familiar with, okay? Some of them are a little bit touchy, but keep in mind, I'm not asking you to memorize all of the little details about it, but you have crude mortality rates, general death rate in a population. And you don't determine it on any particular cause of death. Cause-specific mortality rate, this is where you measure deaths due to a particular cause in a given population during a specified period. Infant mortality rate, death rate of children under age 1 as compared to the number of live births. Maternal mortality rate, the maternal deaths per 100,000 live births for any cause related to pregnancy or management of the pregnancy. And finally, the case fatality rate. This is the percentage of people with a particular diagnosis who die in a specified time period after the diagnosis. Sounds like we're getting picky, but think about this. It's one thing to sit there and say, oh, okay, let's go back to Ignaz Semmelweis. The maternal mortality rate was fairly high. For all these doctors that delivered the babies, and the mothers would get basically a septic infection afterwards. Till he did what? Had them wash their hands prior to delivering babies. And he compared the maternal mortality rate of the doctors to the maternal mortality rate of the mothers that had their babies delivered by midwives. Big difference. Now, you want to also understand that epidemiological studies can be categorized as either descriptive or analytical. Descriptive epidemiology, it uncovers who is infected, where the cases go, when the cases occur. The goal is to describe the occurrence and distribution of the disease. Correlational studies, otherwise known as ecological studies, they search for possible associations between exposure and the development of a disease in a population. And then, of course, there are some other studies that basically are called case reports. This is where an individual or group records, basically has records of a disease. For example, in something like this, you would say, all right, we had this population of individuals that went to a biotech conference. Some of them got infected. They all occurred in Boston at a conference. And when their cases showed up, when they popped positive for COVID-19, they were in these other cities. And as a result, they helped lead to the distribution of the disease. With correlational studies, you try to make a possible association between exposure and the development of the disease in a population. So it might be the situation where the individuals or several individuals were exposed, but they all seem to have had the disease in the same place, a nursing home. Whereas you have case reports, individual or group records of the disease, where the individuals, let's say children, start having unique symptoms that's associated with having COVID, but it gets far worse for these individuals. Now, we also have what we call cross-sectional studies. These are also known as prevalence studies. They evaluate exposure and the development of a disease across a defined population at a single point in time. Often, the use of these surveys has been done to determine the prevalence of the disease. Analytical epidemiology investigates what caused the disease, the etiological agent, and why people get the disease, and how the disease can be prevented or treated. The observational studies do not involve treatment or intervention, but merely watch and collect data on the cases that exist, used to exist, or developed over time in tracked or monitored groups. The last one, to help you understand that, think about this. It's basically, do they get it or not? And if they did get it, maybe this is older data, but we know that they got it, etc. But we don't care about how they were treated afterwards, whether they lived or died, or whether they walked out of the hospital. Okay, so that would be observational studies. It is experimental studies that permit the researchers to change of variables and to determine the effect of the change in the outcome. A lot of times this can include also clinical trials. So experimental studies, we have a bunch of COVID patients. We will try them with hydroxychloroquine, the chloroquine, and maybe we'll add zinc or azithromycin. And these are the clinical trials to see if there is a change in the outcome. Do the patients get better? How fast do they? Remdesivir, maybe what it is that it doesn't cure them, but it reduces the outcome in the sense of reducing the time that they're in the bed before they're able to recover. So there's a whole mess of experimental studies that occur that kind of go hand in hand with clinical trials in an attempt to control the disease once the person has it. Now, here's an example here. We have cervical cancer patients studied. 85% showed the evidence of human papilloma infection. 15 showed no sign of this. So this study, which basically took all of these individuals, found that there were some that had HPV, the virus, but very few did not have it. Now that raises questions. Are there other factors or other agents that might initiate the cervical cancer? So you can get a lot of questions started as well as answered in some of these studies. Here we have maternal mortality rate. Okay, so this is the stuff we talked about a few minutes ago with Semmelweis. Ignaz Semmelweis was sitting there going, wow, look at the death count here, the percentage of deaths of the moms after they have delivered their children. Compare that to midwives. And in the midwife wards, they were virtually very, very small. Once they started implementing hand washing in the physician's ward, you notice this plummeting amount of cases of maternal mortality. And it basically almost then shadows the midwife's ward throughout the rest of this decade. And that brings me to a little bit more information here. I want you to be aware of when we have clinical studies, sometimes what we do is not just merely do we give them a drug, do we give them a vaccine, etc., but is some of the strategy or some of the processes that we do in a hospital setting, in a healthcare setting, may actually enhance the spread of diseases or limit the spread of diseases. 1840s, Ignaz Semmelweis first showed that hand washing prevents disease. We have a lot of what are referred to as healthcare acquired infections, HAIs. They are dangerous, expensive, and an increasing problem. And that's why patients in some cases don't want to stay in hospitals as much. I don't want to get something else. Now, we formerly used the term nosocomial infections when we talked about this. But there are a variety of strategies that infection control specialists are working on. in healthcare facilities, not just hospitals, but also nursing homes, etc., to try to limit or actually reduce incidence of HAIs in these facilities. And you got to keep in mind that when we talk about healthcare epidemiology, some of the simplest things, preventing contamination from contaminated hands or medical devices, that's key. Bacteria are the leading cause of healthcare-associated infections. Now, common HAIs, this is an extension on this, but I want you to keep this in mind. These can come from contaminated medical devices, healthcare worker hands. They can be direct or indirect sources. They can be localized, restricted to specific body part, or systemic, affect any body system or tissue. For example, we have such pathogens as Clostridium difficile, terrible, C. diff, as some healthcare workers have told me, and it's a mess to treat. We have MRSA, now methicillin-resistant Staphylococcus aureus. This is basically an antibiotic-resistant form of Staph aureus. Very difficult to treat. Escherichia coli, Pseudomonas aeruginosa. Pseudomonas P. aeruginosa is very common as an HAI, and this really takes concerted efforts to prevent contamination and where they see it to work on eradication and decontamination as quickly as possible. You see, the role of healthcare epidemiology programs leads to savings that are measured in not only dollars, but in lives. So how do we prevent HAIs? And this is kind of going back to aseptic technique, but this is more specialized here for someone that walks into a healthcare facility. Personal protective equipment, PPEs, gloves, mask, goggles. Remember, even COVID can be transmitted by some of the airborne particles that have the virus, but instead of not being breathed in because they're blocked by the mask, they come in contact with the eye, and there they have their portal of entry. Hand wash, environmental sanitation. This includes also regular prompt cleaning of facilities that are contaminated, as well as prompt disposal of all contaminated materials, okay? And of course, equipment sterilization. So that includes all the tools, all the endoscopes, and all the surgical materials, etc. Now, hospital epidemiology and superbug HAIs are a real threat today. Patient safety is threatened by the antibiotic-resistant strains of HAI. This requires infection control teams to devise strategies to prevent the transmission and make informed decisions on antibiotic usage. Look at the vast amount of infections caused just by C. diff, over 55%. Now, here's an example of an HAI device that becomes problematic. It's a catheter-associated urinary tract infection, a UTI that's caused in part by the catheter. A lot of times what plays a role in that catheter-associated UTI is the organisms and how they form a biofilm. And the biofilm, remember, protects the microbes inside from disinfectants and from antibiotics and things like that. So a lot of times it's a matter of you have to just keep removing the catheter and putting in new ones to prevent the resurgence of the biofilm and prevent the individual from getting an HAI. Sometimes these infections, not a lot, but still potentially present, at 11% is the surgical site of where you have the infection. Others are the central line associated bloodstream infections. Central line associated, translated, you have indwelling basically venous catheters, things like that. And also we have MRSA bacteremia. Now, don't worry, I'm not asking you to memorize all of these different little nasty HAI pathogens. Just take time to review some of them so that you clearly understand that you can have bacteria, you can have viruses as HAIs, and even fungi. And some of the fungi are getting more and more difficult to control because there's now, for example, the Candida aureus, which has been found to have multi-antifungal drug resistance, and that's going to be a major problem. Now, we've got to bring down a few more points as we finish up here. Surveillance, eradication, and ethics in epidemiology. There are a series of notifiable and reportable diseases. Actually, Congress funds that state agencies report to sort of a central distribution about 40 different communicable diseases. But that's not all the communicable diseases that are out there. And it certainly doesn't give you a full picture of health care or the costs involved to health care related institutions. Okay? The CDC has a national notifiable disease surveillance system, the NNDSS. This is a network that depends on state and local hospitals, laboratories, and private healthcare providers to monitor and report cases of specific diseases. Therefore, you have situations if they have a sudden, surprising, nasty disease, such as, oh, plague. Plague? Yeah, plague. You mean like the Black Plague? Sure. They had a time where they were showing up because the fleas that carry the plague do not care whether they're on puppy dogs or prairie dogs. They have been found that prairie dogs can have some of the plague bacteria. Of course, they're marmots if you go to the Central Asian area, and they're very common to have those fleas with that type of pathogen. Okay, Yersinia pestis, which is the pathogen that causes plague. So anything that's really seriously reportable has to be brought to the notice of CDC. That information may be compiled, and they'll just sit there and say, okay, does this mean we have a trend? Do we have an outbreak? Do we have just an isolated case or two? What? Reportable diseases, these are diseases on state or local tracking lists. And we also have notifiable diseases. The CDC has a list of 60 infectious, and some of them are non-infectious diseases that they have to monitor, and everybody is supposed to report. Now, here's another document that if you are interested, you can easily get on the web, and you can actually get it delivered to you each week. It's called Morbidity and Mortality Weekly Report, MMWR. It's a publication from CDC. Basically, it summarizes health watches and update reports of selectable notifiable diseases. Yes, you'll get information on COVID. Yes, you'll get information on HIV. Yes, you'll also get information on the unusual outbreak of the deadly E. coli version, the O1 or H17, etc. All of the different types that pop up, and some that are unusual, may be reported in the MMWR. For the very first situation that occurred with HIV, it was a report in the MMWR of a small cohort, that is a small group of gay men that started showing up with Kaposi's sarcoma in June of 1981. That was considered sort of like the benchmark or the herald of this is what we've got facing. Now that was 1981. The attention for HIV did not build up until we started looking at 83, 84, 85, 87, and 89. Okay? But this sort of unusual case of this very, very rare cancer in this small group population was what was the key to get investigators to start looking at something here. Now, this is basically a nice diagram. It's at the end of the chapter, and it's part of the review, but it's important that you keep this in mind. What's key to global health, if you really want to say it that way, is reporting. The local, regional, state public health authorities pass this information. It comes from laboratories, healthcare providers, hospitals. This may be then moved up the chain of communication to the national authority, which is the CDC. This may also be passed up to the global authority, and of course, its concerns are somewhat voluntary, and that is the World Health Organization. Now, when we talk about surveillance of emerging and re-emerging diseases, an emerging disease is a new or newly described infection in a population. You got a bunch of them, okay? SARS, which is the severe acquired respiratory syndrome, Ebola, COVID-19, HIV, swine flu, these are all examples. Excuse me. Re-emerging diseases, though, are diseases that previously were under control but now show increased incidence. Drug-resistant strains of bacteria, cholera, plague, each one of these has popped up. And I'll just share with you really a quick thought here. Years ago, I used to get these, you'd get these basically these almanacs for the year. And they would have all sorts of information in them. And I remember one of them that I had kept for a while. And you turned a certain series of pages and it had disease outbreaks over the centuries. And when you started noticing this, and this is also what further helped this accumulation of information was the fact that the world was starting to communicate one end to the other. And not just from a historical perspective, but actual real time. So whether it's by newspapers, by radio, or whatever. And every once in a while you would have outbreaks such as cholera or smallpox. And when we had an outbreak, it wasn't just a couple of hundred people dying. It was 1 million died, 5 million died. And these were incredible outbreaks. But eventually, public health, epidemiologists, etc., were able to start pushing back on this. Unfortunately, from time to time, we start getting, I'm going to just say it nicely, sloppy. And sooner or later, certain diseases start popping back up. Here's one example. Sadly, in some of the communities around, I believe it was Oakland, Los Angeles, and San Francisco, because there was such a burdening amount of homeless and population, many of the homeless were not able to properly have access to bathroom facilities. So they would empty their bowels right on the street. And over a period of time, this led to outbreaks of hepatitis A. And you think to yourself, wait a minute, this is the United States of America. We have all sorts of treatments, etc. How is Hep A occurring? Because of public sanitation that has fallen through with maintaining cleanliness on public streets. Just happens. Now, here's an example if you want to take a look at some of the diseases that are out there. MERS is Middle East Respiratory Syndrome, similar to SARS. And basically, the reservoir for MERS, COVID, and SARS has been what is believed to be bats. Ebola, the reservoir is believed to be bats also. Lassa virus is a type of rat. The Hendra virus was bats also, as was the Nipah virus. The difference with the Nipah, though, was it went from pig to human and then eventually was able to achieve human-to-human contact. And this was basically, in some cases, by ecological encroachment, meaning that human populations would come closer and closer into areas that was primarily basically the environment for various zoological species, vertebrates, invertebrates, etc. And with West Nile, birds, mainly the birds that were migratory, and some of them went across the United States, and this was West Nile virus. So you can see how some of these things can occur. Same thing here with avian flu, wild birds. The transmission, although it starts somewhere out in Eastern Asia, can literally travel thousands of miles by the migratory birds, which makes it very interesting. Okay, so how do we deal with this? Well, eradication is the ultimate triumph over an infectious disease. When I talk about eradication, this is an infectious disease that is there are no longer any cases anywhere in the world. And you have to keep in mind that there are only certain factors that make eradication possible. You have to know if there's a reservoir. You have to exterminate the presence in the reservoir. You have to easily identify the pathogen, have treatable, preventable infectious diseases that only humans can transmit and catch. Perfect example, smallpox. They spent millions over several decades. And eventually, because of this pursuit of vaccination of populations and development of herd immunity, the world doesn't have smallpox anymore. Unfortunately, there are some individuals that may want to see smallpox reemerge, particularly those that favor bioterrorism. It's a sad thought, but it does exist. There's only samples that are known to exist in the Centers for Disease Control and Vector, which is the Russian site for viral research. The reality was that the Russians had violated the Bioweapons Treaty of 1973 and for 20 years had been mass producing smallpox without letting anyone know, with the idea of being able to put it on the warheads of ballistic missiles and let it rain down on enemy nations so that you would have a large amount of the population exterminated via smallpox. As there are no other basically reservoirs for smallpox virus, except for those that are in samples in some research labs, there is no longer smallpox on the planet. There are several other attempts for eradication. One of them is being targeted right now globally for polio. Another is the guinea worm, okay? And that is a parasitic worm that can lead to blindness, etc. Those are still in the intermediate stages of success, but they're making headway with those diseases as well. We have to keep in mind the ethical considerations and vaccinations. There is a lot on the fact that past vaccinations have saved lives. Unfortunately, there was a series of several individuals, mostly one of them, who developed a pseudoscience, a false paper, that led to suspicion that vaccinations resulted in children getting autism. As a result, individuals jumped on this and they thought that autism, it may be poisoning, it may be a religious bias or adverse reactions. And the adverse reactions are exceedingly low in any vaccination approach. But the individuals that still herald this pseudoscience being anti-vaccine, to this day, still uses the example of that particular researcher's paper, even though it's been renounced and denounced by all sorts of scientific communities. The gentleman that had authored it had to basically give up his career because he had basically created a false scientific report. Now, herd immunity. You probably heard a lot about that, but let's just give it to you straight. This is the level of immunized members of a population which result in the pathogen that does not have enough susceptible people in the community to sustain the infection. Usually, herd immunity has to be up in the 75, 85, 90 percentile of the population. So roughly those numbers of the population that are vaccinated to basically suppress any real breakout of any of these diseases. Now keep in mind, we're talking about measles, mumps, German measles. We're talking about diphtheria. We're talking about rubella. We're talking about polio. And a lot of people have been so put aside, they have been so divorced from seeing these actual incidents that they don't really have a clear view or an understanding of what some of these diseases will actually do to a person who gets these diseases. Now, there's a concept called R naught. It looks like an R with a 0. It's a mathematical term that indicates how contagious an infectious disease is. Now, it's referred to as also the reproduction number. As an infection spreads to new people, it reproduces itself. So the R0 or R naught tells you the average number of people who will catch a disease from one contagious person. Now, there's some nasty diseases out there that have an R naught of 18. That basically means that it's a highly contagious disease and that people who are not vaccinated, who are susceptible to the disease, everybody around is going to get it or a high risk of them getting it. Basically, R naught is important and it specifically applies to a population of people who were previously free of infection, thereby they're native, they have no immune defense, and they haven't been vaccinated against the disease. Now, this is the other problem. If you have no one there that has any immunity against it in the community, that means that everybody in the community is susceptible. Now, there are 3 possibilities that exist for the potential spread or decline of a disease, depending on its R naught value. If R naught is less than 1, that basically means that each existing infection causes less than 1 new infection, and sooner and very quickly, the disease is going to decline and eventually die out. If R naught equals 1, each existing infection causes 1 new infection. Basically, the disease will stay alive and stable, but there won't be this outbreak or epidemic. If R naught is more than 1, each existing infection causes more than 1 new infection. The disease will spread between people, and there may be an outbreak or an epidemic. Now, as I said, the diseases R naught only applies to everyone in the population that is completely vulnerable to the disease. Nobody's been exposed previously, so they have no immunity to it. Nobody's been vaccinated. And there's also no way to control the spread of the disease. So when you take a look at something like this chart, this is an R naught chart, okay? It's a mathematical term that indicates how contagious an infectious disease is. So you look at mumps, you look at polio, you look at smallpox, you see that roughly every infected person comes in contact, they could spread this to anywhere from 4 to 7 people. When you get to pertussis and measles, it's even nastier because there the R naught is much higher, 12 to 17, 12 to 18. And let me give you one other little factor that came out in a paper relatively recently. It was found that when measles attacks someone and they get sick, what measles, interestingly, does is it wipes out the immune memory of the patient's prior immune responses to other pathogens. It's like going up to the immune system and saying, I'm wiping you totally clean. But it's not a good clean. Now you have no memory against other diseases, and therefore you're susceptible to them. So measles is a devastating disease. If you survive it, okay, but if you survive it, you may now become very susceptible to a variety of other diseases. Now, you see the threshold percentage? That's basically the herd immunity there. For you to really be able to quench an outbreak of, let's say, mumps, polio, etc., you've got to have somewhere in the range of 75 to 86% of the patients, of the population, I should say, vaccinated against mumps, 80 to 86 for polio, smallpox, 80 to 85, diphtheria, 85, rubella, 83 to 85, and look at pertussis. 92 to 94%. Measles, 83 to 94%. Now, I want to throw out to you one other thing, and it's not me getting on a soapbox. Do what you want. But here's something to think about. We have had, in the past 2 decades, I've watched news reports where certain large populations, communities like London or Birmingham or Minneapolis, the herd immunity has dropped down below those thresholds. So it's just a matter of time before, oops, there's an outbreak of whooping cough. Oops, there's an outbreak of measles. Oops, there's an outbreak of rubella. And part of this is because of, yes, the anti-vaccine sentiment, but the other part is this. You see, many of my students right now listening to this recording are now in your late teens, early 20s, mid-20s. When's the last time you had your vaccination? Now, if you're nursing students, you probably had to re-up a bunch of your vaccinations. When I started teaching microbiology, I started to realize, oh my goodness, I was so many years older gentleman, and I hadn't had a booster in a long time. Once you get measles, the vaccine, the problem was a lot of adults, as they walk into their 20s and 30s, start going, well, I used to do this when I was a kid, so I should be immune. Wrong. You got to get a booster. In some cases, it's a booster every 7 to 10 years. And if you get that, then you're continuing to maintain your immunological memory and your immunological defense against these diseases. But there are a lot of cases where, let's just take tetanus, for example. The incidence of tetanus would increase in the age groupings when you go from 30, 40, 50, 60. And when I mean increase, it's a really steep slope as you go from 40 to 60. Reason? Unless people had a follow-up vaccination, which they need every about 7 to 10 years, they don't have any protection against tetanus later on. And it's interesting that a lot of construction workers were suddenly having outbreaks of tetanus later on in their life, and that's because they lost the immune protection that would normally be provided by a vaccination. They hadn't had a vaccine since they were kids or late teens or something. So this is an extremely important issue to keep in mind. Okay, we're done here. So let's go over a few points. You want to review the visual summary on page 292. Review the chapter overview on page 293. Review the end of the chapter questions on pages 295 to 296. You have in Mastering Microbiology to review the homework as well as the quiz for this week. You need to peruse the videos in the Blackboard for the lecture content. Our next section will cover host microbe interactions and pathogenesis, which is chapter 10. And have a great day.